Good morning, everybody. Thank you for coming. My name's Heidi Feldman. I'm the Division Chief of Developmental Behavioral Pediatrics, and it's my distinct pleasure to introduce you to this morning's speaker, Jason Yeatman. So Jason, I think, could be described as both a pioneer and a star in the emerging field of educational neuroscience. So this is a field, a synthetic field, that brings together education, psychology, cognitive neuroscience, developmental cognitive neuroscience, technology, and related fields, all in an effort to improve teaching and learning for children. And Jason has built, in a pretty short time, an incredible portfolio in this field. So he has contributed to the literature on acquiring and analyzing advanced neuroimaging studies to help with understanding, learning, and reading. He's actually contributed to anatomy through the use of some advanced neuroimaging techniques. He's focused on reading, and he's described the neurobiology of reading, and as you'll hear today, the effects of training on that neurobiology. So Jason received a bachelor's degree from University of California, San Diego, in political science, but as he was coming to the end of his undergraduate days, he started getting a hankering for psychology. Then he got a PhD here at Stanford in the psychology department under the tutelage of Brian Wandel. And then he moved to the University of Washington in Seattle, where he was both a research scientist and then an assistant professor in the Institute of Brain Sciences and Learning, and also communication disorders. So we were able to recruit him back to Stanford with what I think will be a really exciting and distinctive position. So he's half-time here in the School of Medicine, Department of Pediatrics, and Division of Developmental Behavioral Pediatrics, and half-time in the Graduate School of Education. And we're hoping that by fusing his position, he will be on a very solid foundation for continuing to make amazing contributions to the literature, to train the next generation of researchers and interprofessional or transdisciplinary professionals, and also to disseminate a lot of the findings to clinicians and educators. So I want to end this introduction with just a brief personal note. So I met Jason just about exactly 13 years ago. I was a brand new professor here at Stanford. I was just setting up a laboratory on the neurobiology of outcomes for children born preterm with a focus on language and reading. And I needed a lab manager and research coordinator, and Jason applied. And when I met him, I was immediately impressed with his intelligence and his enthusiasm and a kind of boyish wonder about science. I was a little bit worried about his commitment because he had brought his surfboard to the interview and he was planning on catching a few waves on the way back from Palo Alto to San Francisco and Pacifica. It turned out that my worries were not with any standing. We had an incredibly productive 2-year period in which he really contributed amazingly to my research project here at Stanford. And so it gives me such pleasure and pride to think that I had even a tiny role in propelling this extraordinary career. So please welcome Jason Yeatman.
Thank you so much for the wonderful introduction. When she was telling that story, I thought it was a hyperbole, and I actually just thought back and remembered that I did, in fact, actually have my surfboard in the car when I came to interview for a job here. But I will say, when I came back to interview for a job the second time, 13 years later, about a year ago, I did not actually have a surfboard in my car with me. So my lab does research at the intersection of education and neuroscience, and one of the goals of this talk is to introduce the types of things my lab is doing here and hopefully inspire new collaborations. I think I learned I'm actually the first joint appointment between the Graduate School of Education, and the School of Medicine. And that's, I think, opens up a lot of exciting possibilities for building truly interdisciplinary programs. And there's so much shared interest between these schools. And I'm hoping that over the coming years that there will be new opportunities for collaborations that do more than any of us could do on our own. So I'm gonna, maybe this is somewhat unconventional over here, but I'm gonna invite you to raise your hand and interrupt me. If you have a question or a comment during the talk, I might retake the control though, if that gets too much. There's some laughing, so maybe that won't become too much, but feel free to raise your hand if you have a question or comment or anything. And I'll also just say one other thing. So the last time I was in this room, I actually just had a baby upstairs here. That was my first introduction to this side of the hospital. So a month ago, I was in this room getting the introduction to the birthing suite here. So it's kind of a exciting pleasure being back in a different context. Okay. So for those that don't spend their days contemplating literacy, I want you all to appreciate what is unique about literacy in the context of development. So looking at this image here, someone told me I should have replaced this with my younger daughter. This is my older daughter from about a year ago. The two people in this image see fundamentally different things and will continue to for many years. So I look at this page and I can't help but hearing the sounds of language, hearing the sounds that someone else wrote down in written language, while my daughter sees a series of beautiful pictures and hears the sound of my voice, but the text means almost nothing to her.
What we should really be seeing is the slides. Yes.
Oh. Oh, on Zoom, okay. Yeah. And as I said, we'll continue to appear. This is a slightly more recent picture of my daughter where she can now speak, she can interact, she can understand what I say, she can say, Dada, read me another book, but still looking at the page that the text she's looking at means nothing to her. So another important point to keep in mind is that literacy was invented by modern societies. Our brains aren't pre-wired for literacy. No matter how many books are left piled in front of my daughter, she'll never learn to read on her own without systematic instruction in the rules of written language. This is in stark contrast to speech perception, where I never had to teach my daughter how to move her articulator muscles to produce the sounds of our language. That's just something that she picked up naturally from being bathed in a rich language environment. So the point to take away is that the neural circuitry of literacy is constructed in response to education. And there's a couple interesting points here, and for the sake of my research, I think that literacy is this model system for understanding plasticity in the human brain, that we can do many interesting manipulations of the learning environment and actually measure how that shapes the development of a child's brain. So to illustrate this last point, I want to compare what's going on in my brain versus my daughter's brain as we're staring at this book together. So when our eyes move to the first picture in the image, signals are sent from our retina to the very back of our brain, our primary visual cortex, that's involved in extracting the simplest features from this visual image. So recognizing the location and orientation of the line segments that, for example, make up this drawing of a rabbit. Now, as we stare at this image for a couple hundred milliseconds, signals propagate up the ventral surface of both of our brains. This is a broad circuit that's involved in object recognition. And in many ways, this process unfolds almost identically in my brain and my 2-year-old daughter's brain, whereby through signals moving through the circuit, we recognize that we're staring at a picture of a bunny rabbit here. However, things are fundamentally different as soon as I move my eyes to the first piece of text on this page. Within this broad circuit for object recognition, within my brain, there's a specific area that's involved in the rapid and automatic recognition of words. And from this word recognition region, signals are immediately propagating into my speech perception circuitry. So in my brain, you can think about it that through the process of becoming literate, that there's this automatic connection between my visual system and the system that evolved for speech and language. Well, of course, in my daughter's brain, that doesn't exist yet. But as soon as I am reading out loud to her, of course, I'm not just turning the pages, I'm reading this book to her. Now these same circuits for language comprehension become active as soon as she hears spoken language. So as we think about this information flow through the brain during reading, one of the critical things to take away is the importance of connectivity for literacy function, that the physical connections, the white matter tracks that connect different components of this circuit play a critical role in how children learn to read. So from the literature, there's 2 specific tracks that have played a dominant role. The first is known as the arcuate fasciculus. This is a connection between Broca's and Wernicke's area. It's involved in language broadly and also phonological processing, the extraction of phonemes from speech, which is critical for reading. And the inferior longitudinal fasciculus, which is a major output of the visual system, allowing for signals from visual cortex to propagate up to the rest of the brain, and is critically involved in the visual aspects of reading. So if nothing else, there's 4 points I want you to remember throughout the rest of this talk. First is that literacy prompts the development of specialized circuits in visual cortex that are involved in rapid and automatic word recognition. This is one of the largest changes that occurs as children become literate. Second, in the literate brain, written words are processed like spoken words. So you can think of this as the visual system being plugged into our language system in a new way, allowing these two different modalities to be associated together. The third, the structural connections between vision and language are critical. That's going to be a big focus of this talk, is looking at changes in the white matter as children learn to read. And finally, the development of this circuit goes awry in roughly 10% of children. And this is a disorder or a learning disability called dyslexia that involves difficulties in the process of learning to read. So my lab's general approach starts with studies that look like many other educational studies. Usually a small group or individualized instruction program, usually in literacy. But we use these intervention programs as a tool to understand how education or interventions that we're doing are shaping brain development. So over the course of these intervention programs, we're designing and collecting neuroimaging measures to understand how changes we make to a child's environment shape the development of specialized brain circuits. Now, the long-term goal, and this is one of the reasons I was excited to come back here to Stanford, is thinking about how over the course of these intervention programs, as we're collecting these neuroimaging measures, how we can also use neuroscience as a tool to predict and understand and hopefully personalize the interventions that we're performing. So using neuroimaging as a way to understand individual differences in how children learn, and hopefully tailor and time our interventions to the unique characteristics of that individual. So the idea of personalized medicine has been growing in excitement for a while. I'd say the idea of personalized education is a nascent field that is just starting to gain some traction. Okay, so the outline for the rest of this talk. First, I'm going to begin by discussing plasticity in the reading circuitry. And we're going to ask the question of how much does a targeted intervention program change white matter structure in children with dyslexia? Second, based on some of the same data, we're going to examine the idea of whether there is a sensitive period for white matter plasticity. So we're going to ask the question, does plasticity in learning diminish with age? How critical is it that we intervene at a specific period in time, or does this circuit remain malleable to environmental influences for a longer time than we might predict? And finally, I'm probably not going to get to this, but if there's time at the end, I'm going to be talking about new work in my lab, looking at establishing the foundations of literacy, where we're going to be doing intervention programs in pre-literate preschool children and looking at how that shapes development over the long term, and asking the question of whether establishing a solid foundation helps prevent dyslexia down the road. Okay. So to dive into the first part. So for those that aren't familiar with dyslexia, dyslexia is typically defined in terms of neurobiology. This is a definition I pulled from the International Dyslexia Association website. It was the outcome of an NIH panel focused on coming to a consensus definition. So dyslexia is a specific learning disability that is neurobiological in origin, is characterized by difficulties with accurate or fluid word recognition, and by poor spelling and decoding skills. So behaviorally, dyslexia is observed as children that are having extreme struggles learning to read, learning to automate the process of word recognition, and that this is grounded in neurobiology as opposed to not receiving adequate instruction or lack of motivation. So this definition is also often interpreted to imply that children with dyslexia have a neural defect that causes persistent difficulties with reading, irrespective of intervention efforts. This has been a perspective that's emerged since this definition came out. And unfortunately, I would say that there's a wealth of research on differences in the dyslexic brain, on how it differs from the typically developing brain. But there's much less information on the relative stability or plasticity of these differences. So while we can chart out reproducible findings of what is different in terms of, for example, the underlying white matter connectivity, very little is known about how stable these differences are when we intervene. And that's been the focus of my lab's work for the last 5 years. So we can ask, first off, what is different about the dyslexic brain? And to summarize a vast literature in about 1 minute, one of the critical things that's emerged from the literature comparing struggling readers to typically developing readers is a focus on the white matter connections in the brain. So there's one finding, this is from data my lab collected relatively recently. There's these two white matter tracks, the properties we measure with diffusion MRI, and I'm going to unpack these measures a little bit more later on in this talk. We find these measures of white matter structure are correlated with reading skills and differ in children with dyslexia, specifically in these two tracks, but also a collection of other tracks. This is new data from a couple years ago, but it's largely a replication of now almost 20 other studies that have found that white matter properties correlate with reading skills and they differ in children with dyslexia. So when we look at plots like this, one of the questions that comes up is, are white matter properties a static trait that is determining cognitive capacity? So is the reason that some of these children have difficulty with reading is because the structure of these white matter connections are different? So under this model, I think this is typically the model people are thinking about whenever we have a plot that shows a relationship between an anatomical property of the brain and a behavioral measure, is that children at different points in this plot show different developmental trajectories that are relatively stable over the course of development. However, we could also posit a different model, and we could ask, do white matter properties dynamically change to reflect the ongoing influence of education or experience, the richness of a child's environment? So under this model, we might posit that a child is developing early on in life, and as factors change in their environment, for example, they go to a good preschool, maybe that changes the developmental trajectory slightly. They go to kindergarten, and as they, for example, end up in a classroom where they're not engaged and they don't connect with their teacher and they feel as an outsider of the classroom, maybe the development of the white matter starts going the opposite direction, then maybe they change schools and enter an exciting learning environment for the first time, and that rapidly changes the developmental trajectory of the white matter. So under this model, what we measure at a given time point might reflect a child's immediate environment as opposed to something intrinsically different or something statically different about their brain. Now, of course, this shouldn't be taken as an either or. These are two, extreme sides of how stable versus how plastic are these circuits. But the focus of the present study that I'm going to introduce is looking at this exact effect here, taking children who have dyslexia, who have been struggling in school for a number of years, who have fallen a number of grade levels behind, and then taking them into an intensive summer program where we can basically provide them what we think is like the best possible literacy instruction, and measuring how that change in environment affects the development of these specific white matter tracks. So we're going to ask the question, can we influence white matter development through a targeted educational intervention? And first, I want to thank all the folks in my lab. This is all done at the University of Washington. Doing a project like this involved collecting 562 behavioral sessions, 250 MRI sessions. This is all done in young children longitudinally over the course of a tightly controlled intervention. And Emily Kubota and Patrick Donnelly were the two that coordinated this massive data collection effort. Actually, Emily Kubota is now a grad student at Stanford, and Patrick is finishing up at University of Washington. And then the two postdocs who are heavily involved in developing the analysis pipelines, tools, and that I'm going to be presenting today. Okay, so the study design of this study, we recruited 26 children with dyslexia for an intensive reading intervention program, and I bolded the word intensive to emphasize that this program was 4 hours a day of one-on-one training done over the course of summer, 5 days a week for 8 weeks. These children are ending the school year and then coming in and committing their summer to working one-on-one with a trained instructor in the skill that is the most difficult for them. And the training program isn't just more practice with literacy, it's a systematic build-up beginning with basically reading from scratch, starting with identifying phonemes, so identifying the meaningful units of language from the speech signal, recognizing and visualizing letters and working on associating letters with the sounds of language, building new words from their constituent components, and consistently working towards decoding and rapidly recognizing more complex words automatically. There is also a control group of 18 subjects going through business as usual. This was not a randomized design, and that is one of the limitations. The reason for that is it's hard to randomize when you're asking families to commit this much of their time. Essentially, the families that are coming in are committing their whole summer to doing this intervention program with us. So our control subjects were scanned during business as usual over the course of their typical school year. As part of the study, each subject came in at 2.5-week intervals into my lab, where we made measures of reading accuracy, reading fluency, reading comprehension, and also measures of the structure of their white matter pathways with diffusion MRI. So to show the behavioral data first, this is work by my graduate student, Patrick Donnelly. So this is showing data for one child over the course of the intervention. We'll use that here, see if this works. So the y-axis of this plot is a measure of basic reading skills. This is a measure of the complexity of words that children can decode. It's an age norm standardized measure with a mean of 100, standard deviation of 15. This is a 9-year-old child who is coming in at the floor of this test, which roughly means they're able to decode 3 and maybe 4-letter words. So for any of you that have had a child going through 4th grade, imagine the daily difficulty and the level of stress and anxiety that is characteristic of that child's normal day going to school when they're being asked to read books or read paragraphs in a circle in school, or being sent home with a history book and asked to answer questions of it. That child's only able to read 3 and 4-letter words. This is a child coming in roughly at the floor of the test and showing almost 2 standard deviations of improvement over the course of the intervention. So this was baseline before the intervention, 2 midpoint measures, 160 hours, so after the end of the intervention program. So these are data on other children. These are 5 children randomly grabbed from our sample. The 2 things I want you to appreciate is there's variability in where all the children are starting. There's variability in growth, but all these children are coming in struggling on reading skills, and between the level of kind of slow struggling readers to severely impaired. And there's a lot of variability in growth, but all the children showed improvements over the course of the summer. On average, children go from the 10th to the 30th percentile. This is 1 standard deviation of change, so an effect size of 1, and about 1 grade level to put it in different units. But there is also variability in growth, with some children showing, for example, these 2 that came in at the floor of the test showing almost 3 grade levels of improvement over the summer, then others showing more limited improvement. Yeah.
Can you give some specificity of the entry criteria? What exactly was
Yeah, so the question is, what is the entry criteria? So for all of our studies, we recruit pretty broadly. So the entry criteria for this study is that they had to be a standard deviation below the mean on a certain measure of reading skills, and that their IQ scores and productive and receptive language abilities were within the normal range, that they didn't have any other counterindications, so they didn't have any major neurological or psychiatric disorder. And so my lab, there's a lot of different definitions of dyslexia, and my lab generally recruits pretty broadly, because one of the things we're interested in is what characterizes differences in growth over the course of the intervention. So having a broader sample is actually a strength for us. Thank you. Okay, so we can look in terms of other reading measures as well, and to summarize, basically they show growth on every measure of reading skills. So this is showing a similar plot where we had individual baseline measures during the school year before they came into the study. The red plot is the one that I showed you before. Blue is reading fluency, a timed measure that also has comprehension in it. Green is tower. This is a measure of the automaticity of reading, how quickly and rapidly children can recognize words. So on all these measures, they were either flat or in some cases significantly declining in terms of standardized scores during their typical school year. They came into the intervention and they showed growth over time. We can summarize that growth just by saying what's the average growth rate of these different measures. And the main finding is that there's significant growth on all measures of reading skills. There's larger improvements in measures of accuracy shown in orange than there are in timed measures that require a component of fluency, but there's still growth across both. And There's no improvement in math skills. So the training program was specific to reading abilities. Actually, you can see there's a significant decline in standardized math scores over the summer. Don't read into that as the intervention making math skills worse. These are standardized measures, so if there's no improvement over time, the score goes down, and I can guarantee that none of these children are going home and practicing math skills during their summer vacation after doing a literacy intervention. So this is just the summer slump that would typically be observed. And in the control group, I'm not showing data from the control group, but they were flat on all these measures. Okay. So based on this, we can now get to our key question. So we see large behavioral improvements about an effect size of 1, and we can ask, does the learning process change the structure of the reading circuitry? I'm not going to go into details of the diffusion MRI data. I'm going to try to give a high-level overview of what these measures are indexing, but we collected multi-b-value diffusion MRI data, and we processed it with custom software that my labs developed over the years. And to give you an intuition of what diffusion MRI is measuring, I'm going back to a rendering of this tract, the arcuate fasciculus. Now, this rendering that we make based on diffusion MRI data, if we were to zoom in at a microscopic level, this is showing an electron micrograph of nerve fibers. So there would be tens of thousands of these within each millimeter of this larger white matter tract. And diffusion MRI is measuring the diffusion or movement of water molecules through these nerve fibers. So the power of this measure is that as the organization of the tissue changes, that we're going to see differences in the rate and directionality of water diffusion. So in these simulations, these are showing tubes representing axons with blue water molecules diffusing around them. I've set up these simulations in 2 ways. One to look roughly like a child's brain, where the axons are smaller, more loosely spaced, where there's a lot more room for water to diffuse in between the axons. And over here, in the case of the adult brain, where these axons have grown wider, more myelinated, more densely packed together. And this growth of new tissue creates barriers to the diffusion process, meaning that over the course of development, we observed a decrease in diffusivity. So we see that water diffusion measured with this non-invasive magnetic resonance imaging measure, diffusion MRI, that diffusion decreases over the course of development. And broadly speaking, a decrease in diffusivity can be taken as an increase in the density of tissue membranes blocking the diffusion process. So this data is processed with an automatic pipeline that takes raw data off the scanner, does a series of steps that involve identifying specific white matter connections within an individual's brain, and then extracting these measures of water diffusion to index the organization at each time point for each individual. I'm not going to go through the details of the methods, but for those that are interested in these types of methods, I just wanted to highlight, we just started a new project with my collaborator Ariel Rokem at the University of Washington that involves developing a browser-based, a web browser-based interface for interacting with diffusion data. And I wanted to highlight this here because one of the goals of this project is to create an interface to bring a lot of these methods into the clinic and allow clinicians to easily interrogate and look at the different properties of diffusion in different tracts within an individual's brain. So if these are interested to you, I follow up with an email afterwards. It's a project we're just starting and we're very interested in collaborators. Okay, so diving into the main results. So we're going to focus on this tract, the left arcuate fasciculus first, and we're going to look at diffusion properties in this tract over the course of the intervention. So this bar plot is showing the average mean diffusivity values for all these children when they began the intervention program. So summarized for this connection. And this is showing those measures after 50 hours of intervention. This is about 2.5 weeks. And we already see a significant decrease in mean diffusivity within the first couple weeks of the intervention. And this decrease in mean diffusivity persists or grows a little bit more over the course of the full intervention program. So we see highly significant decreases in mean diffusivity over the time scale of weeks here. When we look at our control group, we see that these measures are almost perfectly flat over time. There is an interaction between group and time, such that there's a significantly larger decrease in diffusivity within the intervention group compared to the control group here. We can summarize these with a little more fine-grained detail, looking along the trajectory of the pathway. So we can slice up this white matter connection from where it enters the frontal lobe, so its Broca's area endpoints, to where it enters the temporal lobe, its Wernicke's area endpoints, and look at diffusion at each time point at slices along the length of the pathway, what we find is that we're not seeing isolated changes in one specific part of the pathway, but a broad change in diffusion over the full length of the connection. This is roughly what would be expected. So we see a significant decrease in mean diffusivity, and this suggests that the intervention is driving the growth of new tissue along the trajectory of the arcuate fasciculus. We can next zoom out and ask, are mechanisms of plasticity spatially localized or distributed? Are we going to find these changes just in one connection, or are we going to see them more broadly over a couple connections that are important for reading? So we're now going to look at the two tracks I introduced at the beginning, the arcuate fasciculus I'm going to show in green, the inferior longitudinal fasciculus, or visual connection, shown in blue. One thing we know about these pathways over the course of typical development, that their developmental trajectories are orthogonal, that some subjects show more rapid development on one or the other, but their developmental trajectories aren't related over the time scale of years. When we look at over the course of the intervention, this is showing data for one subject. In green is showing the trajectory of change for their arcuate fasciculus. In blue is showing the trajectory of change for the inferior longitudinal fasciculus. And you can see, so this is 4 time points, pre, mid, and then post-intervention for one subject. You can appreciate that for this one subject, these two pathways are showing almost perfectly identical trajectories of change over the course of the intervention. Here's showing a subset of 6 subjects from the study, and then the rest of the subjects. What you can appreciate is that there's variability in the timing and rate of plasticity across subjects, but for these two very different pathways that are separated by many centimeters, they show almost identical trajectories of change over time. So we can quantify this by fitting each individual's growth rate for their inferior longitudinal fasciculus and their arcuate fasciculus, and we see a correlation of above 0.8 between the rate of change in each of these pathways across individuals. So someone that's showing rapid intervention-driven change in one pathway is also showing rapid intervention-driven change in the other pathway. So we can zoom out even more and examine what were originally control pathways.
Those two pathways, what's the relative volume difference between them?
The ILF is 1.5 times larger, but there's no overlap in terms of space. There's many pathways in the brain that interdigitate and pass through each other. These two pathways are relatively spatially separated. Yeah. Now, the surprising finding for us, so when we started the study, this, by the way, contradicted all of our hypotheses going in. We started focusing on the arcuate and then secondarily on the inferior longitudinal fasciculus. We expected to measure kind of small but hopefully significant change in each of these pathways. And what we found is quite large change, almost a standard deviation of change in diffusion measures over the course of the intervention, and that these perfectly linked time courses for these two pathways, which is surprising. We've done longitudinal studies where we look over the course of years of development, and you don't see any correlation between the change over time in these two pathways. We zoomed out even farther and examined other pathways that a variety of other pathways, some of which we don't conventionally think of as being involved in reading. Now, what this is showing here, this is a rendering of 12 different pathways in the left hemisphere and also the corpus callosum. And the color map here is showing the p-value indicating the significance of intervention-driven change. So colors going to yellow are pathways showing highly significant change. Green colors mean that there is no change in these pathways over the intervention. What you can immediately appreciate is there's a lot of orange and yellow in the brain, that we're observing significant intervention-driven changes in many white matter pathways. This is showing a plot of how much change in each pathway. And what we see is highly significant growth well beyond the core reading circuitry. So this was showing the plot that I showed before. This is the correlation between growth rates of these two pathways. We can examine this across all these different pathways. And what we find is that there's correlated change throughout an extensive network of tracks. So these colored boxes are showing how correlated the growth rates are for the arcuate and the ILF, or the arcuate and the right hemisphere ILF. And what you can appreciate is there's a collection of pathways that are all showing large change and that are correlated together over time. So the next thing that we can look at is to what extent each individual's time course of change tracks that individual's learning. So I'm going to show a similar plot. This is showing diffusivity values for the inferior longitudinal fasciculus. That was the exact same subject that I showed before. I've taken this subject's reading scores shown in yellow, and I've z-scored them just so we can put them on the same axis. Okay, so mean diffusivity is going down, their reading scores are going up, and anecdotally for this one subject, you can see there's a rapid decrease early on, and then limited change after that, and their time course of change in reading scores roughly parallels that. As we zoom out to other subjects, we see roughly a similar effect, and we can quantify this, showing that within an individual, changes in behavior are occurring in synchrony with changes in the white matter, but the asterisk there is also important that statistically there is a link that as individuals show intervention-driven changes in behavior, we see a related change in the white matter, but there's also a lot of individual variability, and that's one of the goals of our future work is to understand what why some individuals are showing rapid changes compared to others. Okay, so the interim summary so far is that interventions drive large-scale changes in brain structure. These changes occur over rapid timescales, much more rapid than we had hypothesized going into the study, and that these changes occur in synchrony with reading improvements. So for those that are in here who are clinicians and see children with dyslexia or that are struggling with other learning disabilities, another takeaway point is that interventions can be highly effective. If you're worried about a learning disability, I think it's important to make sure that families seek help right away. We've started a website here, dyslexia.stanford.edu, where we're starting to compile resources, and hopefully over the next year or so, that can be a place to send, to look for more information on local resources for dyslexia and interventions. So secondarily, white matter constitutes a highly plastic system that flexibly adapts to environmental demands. That when we see group differences, so if we do a group comparison between two different clinical groups and we see a difference in the white matter, it should not be presumed that this difference is some static underlying trait that differentiates these groups. It very well might be due to differences in the environments that those groups are in. So to quantify this, when we look at the effect sizes in terms of how much change do we see in the white matter over this time scale of weeks, the effect size ranges from 0.5 to 1.0 across the different tracks. And when we compare this to the literature on the size of group differences, when we do static comparisons between, for example, children with dyslexia versus children with typical reading skills, these effect sizes range from about 0.2 to 0.4. So we're actually seeing about double as much change over the course of a summer as you would typically see in a group comparison between groups. And we see this in our study that our intervention group actually overtakes the control group in terms of diffusion properties over this very short period of time. So one hypothesis that comes from this is that maybe the large effects we're seeing are because we're intervening during a specific sensitive period. So maybe there's a sensitive period during which the white matter is particularly plastic and amenable to change, and we happen to be intervening during this perfect period, and that explains why we see such large effects. So I'm going to spend about 5 or 10 minutes showing some data on sensitive periods during the same intervention program, and I'm going to finish up and leave some time for questions at the end. Okay, so we're going to look at the question of whether there's a specific sensitive period. So the age range of this sample ranged from about 7 to 13 years of age. We had a really broad age range here. And this allows us to get at the idea of whether there is individual differences in the amount of plasticity we observe in any individual and the amount of change in reading scores and whether that's related to their age. So this plot here is taken from a review paper by Worker and Hint showing a classic model of a critical period or sensitive period. We have age on the x-axis. You have some measure of plasticity on the y-axis. And the idea is that early on, there's some sort of molecular mechanism that that keeps the circuit stable. The sensitive period opens, allowing it to be amenable to environmental influences. And then at some point in development, other mechanisms come into play to fix the circuit in the state that it's in. So to take a quote from their paper, the classic notion of a critical period is a window, typically early in development, during which a system is open to structuring or restructuring on the basis of input from the environment. So to take a slightly more broad definition for our work, a sensitive period is a time window during which a circuit is particularly malleable to experience. And the hypothesis here is that maybe this sensitive period corresponds to early elementary school, and that we're particularly good at intervening during this specific sensitive period, but maybe less effective after it. So our hypothesis is that the magnitude of intervention-driven plasticity, so the amount of change and diffusivity that we're going to see in each individual subject, depends on how old that subject is. We can detect this statistically with an age-by-time interaction, so showing that there's an interaction between each individual's age and how much change we see over the course of the intervention. And our approach to this study is we developed a formal model of what a sensitive period should look like. We conducted a power analysis and pre-registered that. The pre-registration is up online here. This is actually the first time I've presented this data. The work's unpublished right now. We then collected a replication cohort to make sure that the surprising findings that I just presented hold up in a larger group. And then we just finished analyzing the data very recently. Okay, so to look first at reading skills, this is the same measure that I showed earlier on, and this includes the original intervention cohort as well as this replication cohort. This is our measure of basic reading skills, standardized scores. In purple here is the group average, so this is the same plot that I showed earlier on, but including this new cohort of subjects. And then in blue and red, I've split the group into the relatively older and younger subjects. The older subjects were in middle school and at the end of elementary school. The younger subjects were at the very beginning of elementary school. One thing you can appreciate, so these are standard scores. There's no reason there should be a difference. Just so turned out that the older subjects we recruited were slightly worse or more impaired readers than the younger subjects, but both groups showed the same amount of growth over time. You can see that these two lines are almost perfectly parallel. So statistically, when we model whether we see significant change over the course of the intervention, we have to use exponential notation to denote how significant the amount of change we see is on average. And when we look at the interaction, there's no statistical evidence suggesting there's any difference in the benefit to reading skills between the younger and older subjects. So we can now perform the same analysis for the white matter. In order to summarize white matter plasticity with one measure, we're going to use principal component analysis for dimensionality reduction. This is just a technique to summarize the fact that we see this correlated change. We saw correlated change over a lot of pathways. We're going to use PCA to summarize this change so we can get one measure of the white matter at each time point in each individual. This is showing that average change in our mean diffusivity measure of the white matter over the course of time. So we see a main effect of time. And we can break this into the relatively younger subjects. And this arrow shows how much change there was over time in the younger subjects, the relatively older subjects. And we can compare how much change we see in each group. And there's no support for diminished plasticity with age. So once again, we see no significant age by time interaction. And another way to look at this would be the correlation between each individual's age and how much plasticity we observe in terms of diffusion measures, and there's no relationship. We can zoom out a little bit more and just make sure we're not missing anything, and we can quantify how much change the intervention drives at each point in the white matter. So this is showing a rendering of all the white matter. The color map is showing how much change we see on average during the intervention at each time point. And we can compare that to the age by time interaction. And we basically see no point in the brain except maybe there's a couple points where there may be isolated regions where we see a little bit of relationship between the magnitude of white matter plasticity and a subject's age. But overall, we can see that there's large-scale changes in diffusivity that are not dependent on age. So I want to finish up here and just end with the fact we've done a power analysis ahead of time to make sure that to detect what would be the age range where there would need to be a sensitive period for us to detect it. And our power analysis had suggested that if there was a sensitive period that closed before middle school, that we should have the power to detect it with this study. So the takeaway to the question, is there a sensitive period for white matter plasticity and dyslexia intervention? We can conclude that for the intensive one-on-one intervention program that we employed here, neither white matter plasticity nor reading growth rates diminish substantially between first grade and middle school. And there's a couple important points from this. So I would say, don't use this as evidence to not intervene early. I think it's still really important to intervene as early as possible. Another thing that I could show is the incidence of anxiety disorders, depression, psychiatric symptoms in the older children in this sample who had not received intervention, that the burden that's placed on children by to go through schooling without, as someone with dyslexia and not receiving the intervention they need, that there's many repercussions beyond the fact that they have low reading scores. Okay? So I think it's important to think about intervening as early as we can, irrespective of the fact that interventions are effective in older children. So if you see older children that are still struggling, this shouldn't be taken as like, well, they're past the time window, there's no reason to intervene. Our data suggests into middle school that these interventions are equally effective. We're going to try to extend this age range a little more in future work. Okay, so I'm going to end by introducing our current follow-up project, then I'll conclude and leave some time for questions. So the big surprising finding from the study was both the magnitude of plasticity and also the extent of plasticity, that we see changes not just within what's conventionally been known as the core reading circuitry, but within all these other tracks that I've coded in blue that are not typically considered core parts of the reading circuitry. So the model, so this is showing a schematic summary of our data. So in these two collections of pathways, the core reading circuitry schematized in orange, these other tracks that may be unrelated to reading schematized in blue. Before the intervention, children come in and there's relative stability in both these pathways. They go through our intervention program, and we see this decrease in mean diffusivity for both pathways, irrespective of whether they connect reading-related regions of the brain. And our hypothesis is that our data might reflect an initial phase of learning where the brain is preparing to remodel specific tracks. And under this hypothesis, we might expect that when these children leave the intervention program, so the data I was showing went right up to the last day of the intervention, we scanned them the day they were done, and then they went on back to their normal school year. We actually don't know what happened, whether it could be possible that for many of the tracks, the diffusion properties go back to baseline or they continue growing over time. There's many different possibilities. We just started an R01 when I moved here to Stanford that's getting at this point of what are the long-term effects of a short-term intervention program. And our hypothesis is that automatizing this learned skill depends on spatially localized changes in myelin. So using new neuroimaging techniques, we're trying to relate the short-term learning trajectory to what occurs over time as this process stabilizes itself. More broadly, the goal of this follow-up project is to understand the interplay between changes in the structure of a brain circuit with changes in the computations performed by that circuit with actual learning that we can measure through standardized tests and also through measures of the child's interactions in a learning environment. So that's the project that we're just starting right now. And I think I'm gonna finish up right there and not go into our new work just to leave some time for questions. But want to thank, first of all the various funding sources, the NSF and the NIH in particular, then all the many people in my lab. Actually, this is probably about time to update this picture. This was taken right before we moved from University of Washington down to Stanford here. So thank you all for coming this morning. Yeah.
I'm wondering if you could speak more about the difference in baseline. I see the parallel improvement, but the starting point and ending points are still substantially different. Do you think that's just a recruitment bias?
Yeah, so the question was, so in terms of both reading scores and white matter measures, the older and younger subjects differed at baseline. They showed parallel change, and then they also differed at the endpoint. I think in terms of behavioral scores, it was just a recruitment bias that when we're recruiting for studies like this, so we didn't, in doing the study, we didn't have any different criteria for the older or younger subjects. It was just there was just one enrollment criteria for the study. And it turns out that children that are older that are looking for studies on dyslexia tend to be the ones that are more impaired. So children that are 12 or 13 years old that are kind of at that border between, like, are dyslexic, but they're able to get by and compensate, just aren't, that's our theory. They're not spending as much time looking for this. So there is that confound, and that's one of the things we're hoping to look into more in the future. You could imagine there's some interaction between level of impairment and how much growth, and that ends up being correlated with age. We modeled that statistically, and that didn't seem to be the case, but obviously it is a possibility. In terms of diffusion measures, the differences at baseline just reflect that these measures do change over the course of development too, and that's the expected direction. Yeah.
That's impressive work. You use mean diffusivity to actually identify change. That's impressive. So, but what is actually happening on a neuroanatomical basis? Is this better presentation? Is it good tracks? Is it improved myelination?
Yeah, so the question is that what's the underlying neurobiology? And so mean diffusivity is an incredibly sensitive measure. If your goal is to detect, is there a change in the brain, it's our most reliable and sensitive measure. It's great psychometric properties, it's highly reliable. It's also highly unspecific. It's going to be affected by glial cells, by axons, by myelin, that many different changes in the white matter are going to drive the same measured change in mean diffusivity. So some recent work that's under review now takes more complicated models of the diffusion signal where we can measure diffusion in a number of different ways and then posit a biophysical model relating the underlying biology of the white matter within that tract to modulations in the diffusion signal. And from this model, one of the things we can get is we can separate axon water fraction. So if this is a schematic of axons within an MRI voxel, so here's an axon, here are wraps of myelin around it. Axon water fraction is the measure of how much water is trapped within axons. That gives a rough way of getting at how dense are the axons and myelin in this voxel versus, and separate that, so changes in axons and myelin from changes in the diffusion properties around the axons in this extra axonal space that's filled with, amongst other things, glial cells. So this is showing a map of axon water fraction derived from these data. And one thing you can appreciate is that it looks as we would expect, that we see high axon water fraction in parts of the white matter that are core white matter with densely packed axons. It's also quite noisy. So that's why it's not our first measure we go with. It gives us a more specific idea of whether our changes are in axons and myelin versus in the extra axonal space in terms of probably glial cells. But these measures are noisier. That's the caveat. Though a postdoc in my lab did a lot of work to the point where we get the scan-rescan reliability up to about 0.9. So they're noisier, but scan-rescan reliability of 0.9 is still pretty good. So to go, oh, do I not actually have the data here? I think I should have it. Oh no, I lost my slide. Well, I had a slide to the end where I showed the data for this that I must have left out accidentally. But what we find in doing this modeling that pulls apart the diffusion signal to different components, that we actually see no change in axon water fraction. So it's suggesting that over this rapid time period, we're not actually seeing changes in myelin or the growth of axons in terms of being larger in caliber. What we're probably seeing is changes in the glial cells that are making up this extra axonal space that is consistent with mouse work actually coming out of Stanford, showing that in the early phases of white matter plasticity, you see a proliferation of oligodendrocytes that will eventually go on to create myelin, but might not be doing that during this early period. And that's one of the specifics of the hypothesis for the previous, the study we're working on now. Yeah.
Looking at your fantastic fusion work, it almost looks as if you could use this to diagnose dyslexia on babies.
So there is some work, yes, the question around whether just baseline measures can be used to diagnose. And there is some so work from a lab at Harvard has been doing that, actually showing that these same tracks that showed differences between dyslexic and typical readers in the ages I see them also show differences in 6 to 12-month-old. Though those differences are relatively small, they're not at the level of diagnosis. So a lot of people are hoping that they are, that they eventually will be able to be used for diagnosis. But I think one of the things that we're also realizing is that environmental factors change white matter so much. I mean, this is, over 8 weeks, we're seeing a standard deviation of change within individuals. And that has made me realize that a lot of group comparisons might be picking up on differences in environment. You look at measures of the white matter in, a 2 or 3-year-old child coming from a family that has a family history of dyslexia, you may be picking up on subtle differences in terms of how much that child went through, how rich the language environment, other environmental factors. So I'm not convinced yet that we're actually picking trait-like differences can separate individuals versus a sensitivity to the fact that there's many environments. Though it still might be useful for diagnosis.
Just gonna say, it's only, keep, you're asking, it's more, down some.
Yeah, that's a good one. I mean, yeah, like I would say, what would be the downside is you're just saying, make sure you focus on these skills. Yeah, totally. And I think the other way to think about it is, if you collect a couple of behavior measures and these MRI measures, that together those are going to be the best predictors of long-term development. Developing risk scores. So the browser-based product, that's one of the goals of being able to have a tool where we can take a scan of someone off the scanner, use the models developed from these studies to give, hopefully, a risk score saying, the odds ratio of this child developing disorder is higher. So you should intervene early.
Welcome, Jason. This is phenomenal work. I'm curious about your conceptual model around modifying factors, particularly executive functioning on one hand, and also environmental factors like bilingual environments, and how you're approaching both the external environment and the internal.
Yeah, that's a huge focus in the next study. So in this first study, we were just hoping to be able to measure that. We were shocked how large that we just didn't have all the questionnaires that we should have had those other factors. That's something that we're innovating on the last 3 months. I've been here, been meeting with people, I have no expertise in executive function, but there's a lot of answers that do. Bringing those in and good environment. Another issue I guess, and we're changing children's self-concept around themselves and literacy and moving to threat they feel in school. And a lot of the change might have been driven by social, emotional, interpersonal factors as opposed to the specific aspects of the curriculum. So that's what we're getting at.
Why don't we call the grand rounds over so that everybody can get to clinic? But stick around if you have some additional questions, and let's give Jason a thank you.